fantastic. So today I'm going to tell you about our work defining innate immune inflammatory pathways and their role in contributing to the progression of osteoarthritis. I'll start by just reviewing some of the imaging results that show that synovitis is a predictor both of pain as well as progression of disease. I'll then describe our performance of proteomic analysis, identifying different inflammatory mediators that we then kind of identify potential pathways and use knockout mice to start to address which pathways may actually contribute to pathogenesis. I'm going to present just one slide on complements, one slide on integrins, and then spend a little bit of time talking about our recent results demonstrating that mast cells of all cells play a critical role in mediating the progression of OA. And then just kind of conceptually how my lab and my approach to OA might be moving forward. So as you all know, when you tap the synovial fluid of a patient with OA, there's a low number of inflammatory cells, and histologically, as compared to normal synovium, the layer has hyperplasia, and there's infiltrates of inflammatory cells, but kind of an order of magnitude, generally an order of magnitude less than what's seen in rheumatoid, at least the high inflammatory subtype of rheumatoid. There's been quite significant studies, I think in part started by Michael Felsen at Boston University when he basically gave patients getting MRIs of their knees for OA, he gave them gadolinium. And lo and behold, this is like as recently as 2010, and lo and behold, 2/3 of these patients with garden variety OA have significant synovial enhancement on gadolinium MRI. And further, Peggy and Carlos Sanzello here showed that meniscal injury and meniscectomy was associated with synovial inflammation histologically, and that was also associated with increased pain. And then there's been a series of studies that further demonstrate that that synovial inflammation that they observed and that's detected by this MRI fusion and synovitis is associated clinically with increased pain scores as well as increased rates of cartilage volume loss and progression of OA. So, we really view OA as failure of the joint as an organ system. It involves all the different tissues within the synovial joint, including, the synovial lining, as we looked at histologically a minute ago. There's obviously breakdown of the cartilage. There's extensive remodeling of the bone. And I think all these different kind of tissues that are involved in this pathogenic process and the failure of the joint as an organ contribute in different ways to promote, in my world, inflammation that further contributes to the progression of OA. So we wanted, back kind of 10, 15 years ago, we were collaborating with David Lee at the Brigham, and we wanted to gain further insights into what mechanisms might be involved. And so we did kind of a bottoms-up proteomic survey of the synovial fluids from OA patients because, synovial fluid was accessible, it bathed all these diseased tissues, the synovium, the cartilage, the bone, and thus we thought we could gain insight that way. And the first thing we did was just a simple cytokine profile. And this is just, we got normal, we got some sera here, OA sera, OA synovial fluid, rheumatoid sera and synovial fluid. And you can see this, the red is increased cytokines. This is just a panel of cytokines by Luminex. But lo and behold, OA synovial fluid has elevated classical inflammatory cytokines, not nearly as high as what you see in rheumatoid, but they're present and they're easily detectable. Further, if you look at Kelgan-Lorentz score 3 or 4 OA, so kind of more severe OA, even in the blood, you can detect a signal for certain increased inflammatory cytokines. Working with Connie Chu at Stanford, we've been profiling cytokines in some of her cohorts, including ACL tear and degenerative meniscal tear cohorts. And again, we see, low levels of increased inflammation in subsets of these patients. And it suggests, that not only are these potentially intermediates for the development of OA, but that inflammatory process has already been initiated and may be contributing to the progression of the joint failure. So with David, we did this 2D Dodge proteomic survey. We took 10 healthy synovial fluids, 10 early OA, and 10 late OA synovial fluids. We used mass spectroscopy. We identified 2,000 spots, 260 different proteins. When we organized these by kind of the classical pathway analysis, not surprisingly, the top hit is kind of acute phase response, effectively wound injury, a failure to resolve your wound. It's kind of the top two pathways. And then we started seeing things like coagulation. My lab has been very interested in the intersection between coagulation and inflammation, and the enzymes that regulate coagulation also frequently regulate inflammatory mediators. And we have several stories around that in osteoarthritis. And then immunologic disorders with cytokines and immune cell receptors. Already showed you that there's certain elevations in certain cytokines. I think the data on IL-1 in OA is intriguing, but also confusing. So we'll see what happens there. And then complement was another hit. So based on these results, we started to make these models where we kind of hypothesize what immune pathways might be involved. Perhaps there were TLRs and DAMPs, perhaps some sort of phagocytic mechanism to clear cartilage debris, perhaps aberrant stress signaling or local inflammation. And over the last 15 years, we've taken approximately 100 different strains of knockout mice that are deficient for molecules or receptors or cells related to our proteomic findings. And out of those 100 knockout mice, we probably have 30 phenotypes. Frequently, they're organized in pathways. We also have many negative results. And I'm just going to show you some highlights from those and then how that led us to study mast cells. So first, just one slide on complement, one slide on integrins. We're very interested in complement playing a role in driving inflammation in OA. We've published several papers on this. We think cartilage breakdown results in the release of different cartilage matrix proteins, such as fibromodulin, which we show in our Nature Medicine paper, which activates the complement system. Specifically, it activates the alternative pathway, resulting in the formation of the membrane attack complex. The membrane attack complex classically is used to kind of punch holes in virally infected cells or in bacteria to kill them. But in contrast, in osteoarthritis, perhaps it's killing some chondrocytes, but maybe more importantly, it's sublytically stimulating inflammatory pathways in both chondrocytes and synoviocytes. And we show that in our publications. This sublytic MAC stimulation or activation of the chondrocytes and the synoviocytes results in the production of inflammatory and degradative mediators. These cause further inflammation and tissue injury to the joint, and this kind of resulting in more extracellular matrix release in this kind of feed-forward process. And then just one other, we recently published on a key role for integrin beta-3 and CD47 in osteoarthritis, using both human studies and human tissues as well as mouse. This is the mouse result. This is the don't eat me signal that regulates macrophage clearance of debris. And basically, if you lack the don't eat me signal, you get severe or if you lack the don't eat me signal, you're protected against OA. And if you have the don't eat me signal, then you're more susceptible to OA. And this just shows, working through the different receptors and the signaling molecules that mediate that. So I want to spend the rest of my time talking about mast cells. And this arose due to our initial proteomic survey, in which we found evidence of immune cell activation and perhaps FC activation. And that led us to choose different FC receptor deficient mice to do our OA screen in. We're just taking different knockout mice. We're doing the DMM or medial meniscectomy mouse model. And lo and behold, we found that FC ER1 alpha deficient mice are protected. This is half of their stifle joint, so half of the knee joint. This is Safrano staining. They're protected against the development of OA as compared to wild type mice. And you can see in the blinded scoring that the FCE R1A mice are statistically very significantly protected against OA. And so we started thinking about what cells this specific FC receptor was expressed on, and it's obviously expressed on mast cells. And across from our lab is Steve Galley, who's been the chair of pathology at Stanford, and he's one of the world mast cell experts, and so that led us to start working on these pathways. But in that process, we first just simply took transcriptomic data sets and analyzed them for mast cell activation genes and mast cell associated genes. So here's the FC receptor that we were knocking out, different mast cell chemotaxis mediators, proteolytic processing, proteases, and you can see in OA, all of these mast cell genes and receptors are highly upregulated as compared to healthy joints that did not have radiographic or visual on arthroscopy evidence of osteoarthritis. So there's activation of basically mast cell genes. Next, we looked histologically, and we did stains with tryptase, which is a very specific protein for mast cells. And we found, to our surprise, actually there's, you don't even have to look to find tryptase positive cells, the green cells here, in OA synovium. And this OA is on the right here, and post-trauma non-OA is over here on the left. However, we did not see a statistical difference in the levels of mast cells between these two different, formal, full-blown OA as compared to post-trauma non-OA. And so that got us thinking a lot about, there's no difference in the numbers, but yet, OA is associated with the FC receptor that activates them. And we started kind of reading a lot and thinking more and talking to people about mast cells. And really the question came down to, are these mast cells that we're seeing in synovium, are they just epiphenomena? Are they just there or arriving due to some inflammatory signal and chemotaxing in, or are they in fact pathogenic? And I'm going to show you data in the next kind of 10, 15 slides that is going to argue that they are in fact pathogenic. So the first question we asked was whether these mast cells are actually activated and degranulating in OA synovium. And so we did electron microscopy with anti-tryptase, gold-labeled anti-tryptase antibodies. And you can see basically in the non-OA that the mast cell membranes are intact, whereas in contrast in OA, the membrane of the mast cell is disrupted, and there's tryptase granules outside of the mast cell, suggesting that the mast cell is actively degranulating. In contrast to a neutrophil, when a neutrophil nets, it's a one-time event and the neutrophil's dead. In contrast, mast cells can recharge, degranulate, repeatedly over time. And we did blinded quantitation of the level of mast cell degranulation based on the EM studies, and you can see that in human OA joint tissues, synovium, there's a highly increased mast cell degranulation as compared to the post-traumatic non-OA controls. This wasn't acutely post-traumatic, but months after their trauma when they went in for some surgical procedure and repair. To corroborate this, we performed ELISAs on the activated form of tryptase. So tryptase gets cleaved and activated once it's released from mast cells. So we did this on synovial fluids. And you can see that in osteoarthritis synovial fluid, more than half of patients have significant elevations in this activated tryptase as compared to the post-trauma non-OA. So these data convinced us that not only were mast cells present, but they were actually degranulating and releasing activated tryptase, or releasing tryptase that got cleaved and became activated in the OA joint. And so we next worked with our collaborator, Steve Galley, the mast cell expert, and we did two different mast cell deficient lines. The first was the Kit-Wish mouse, and you can see here that when we take wild-type mice, they get bone-on-bone OA in the stifle joints, and here's the Kit-Wish deficient mouse, and the joint, the cartilage in the joint are largely protected, and we can then re-inject mast cells into the joint to reconstitute it, and if we do so, we restore the OA phenotype. And if we look at, the blinded scoring of cartilage degeneration, the mast cell deficient mice are protected, reduce the osteophytes, and reduce synovitis. So given that basophils are also deficient in this mouse model, we used a second mast cell deficient mouse, the CPA-CRE MCL1 flox mast cell deficient mouse strain. And again, we saw, which is another mast cell deficient mouse. Each model has its own kind of weaknesses and kind of strengths. But again, this second mast cell deficient mouse was also protected against OA as compared to the wild type control and as compared to reconstituting the joint by injecting mast cells into the joint, restored the OA phenotype. And again, on blinded scoring, there's reduced cartilage degeneration as compared to the wild type in the mast cell reconstituted, reduced osteophytes, and reduced synovitis. So we next started to conduct a series of in vitro studies to further understand which mast cell mediators might contribute to OA. And we found that mast cell-derived tryptase can play a key role in breaking down proteoglycan in cartilage. So this is taking human cartilage explants and incubating them with media. You see the proteoglycan layers present. Once you add the mast cell tryptase, it's degraded. You can add a tryptase inhibitor, APC 366, and block it. And this is just scoring of those sections. The addition of tryptase results in proteoglycan release from the cartilage explants, and you can block that release with the inhibitor. We next looked at whether incubation of different cells with tryptase induced inflammatory mediator production, as well as chondrocyte apoptosis, and this is just flow cytometry looking at human OA chondrocytes cultured in vitro. And basically, if you add tryptase, you see, as compared to media in the open bar, you see increased early as well as total apoptosis of the chondrocytes. And further, if you do quantitative PCR and look for expression of different inflammatory cytokines implicated in the pathogenesis of OA, so IL-1 beta, MMP3, and aggrecanase, in all cases, the black bars, the tryptase incubated, there's increased transcriptional expression of IL-1, MMP3, and the aggrecanase and the tryptase treated, and the tryptase inhibitor blocks that increase in all cases. And this is just looking at that cytokine production. Instead of this is by gene expression, and this is by protein, showing the same thing. At the protein level, there's also increase of those same cytokines, IL-1, or a different set of cytokines, IL-1 beta, interferon, and IL-8. So we next asked whether if you block tryptase, so we show in vitro that it seems to be promoting chondrocyte apoptosis, promoting chondrocytes to produce inflammatory and degradative mediators. And so what happens in vivo if you treat a mouse with a tryptase inhibitor and induce it to develop OA by DMM? And we found that mice treated with an APC tryptase inhibitor basically were protected against the development of OA as compared to the control. And on blinded scoring, the tryptase inhibitor mice had reduced cartilage degeneration, osteophyte formation, as well as synovitis. So we were very intrigued by these findings, and we're wondering and thinking hard about how mast cells get activated. And, we're interested in whether perhaps IgE played a role, perhaps certain neuropeptides or other inflammatory cytokines or chemokines played a role. And so we started kind of dissecting mechanisms. And the first thing we did was we took the IgE deficient mouse, and to our surprise, we thought that there was no way that IgE would be involved in OA, but to our surprise, IgE deficient mice are protected against the development of OA. So here's the mouse with wild-type IgE, more cartilage degeneration, and it's generally protected in the IgE deficient knockout mouse. And we also did it with the mouse version of Xolair. So we recombinantly made an antibody against mouse IgE. And we showed that if you give very high doses of this, 1 milligram twice a week, so we're bombing the mice to deplete their IgE. That depletion of IgE also protects mice against the development of OA as compared to treatment with an isotype match control antibody, and this is associated, the IgE depletion, we show that it's associated with complete depletion of IgE in the serum, is reduced cartilage degeneration, reduced osteophytes, and synovitis is also slightly reduced. So we next looked at downstream signaling. Now that we'd implicated IgE, we looked at the signaling pathways through FcR1a. That's the high affinity Fc receptor that we originally identified, that there was a signature in our proteomic screens and that the mouse was protected. So this is the same result I showed you before. The Fc epsilon receptor knockout mouse is protected against OA as compared to the control. And so we looked, this Fc receptor signals through the kinase Syk. And so we used a Syk inhibitor and showed that a Syk inhibitor can protect against the development of OA as compared to a vehicle comparator. And we took, the synovium from these Syk inhibitor-treated mice and basically show that in the Syk inhibitor-treated mice that are protected against OA, there's basically, this is the Syk inhibitor here, this open bar, there's basically a reduction in the transcriptional expression of multiple different cytokines, degradative enzymes, and other inflammatory mediators. So our model is that that there's an IgE-dependent activation of mast cells that plays a key role, we believe in a subset of OA. When you look at our data, 2/3 or so of OA patients have very high levels of this activated tryptase, suggesting mast cell activation. So we would think perhaps that's kind of the proportion of humans where this type of mechanism might play a role. So something about joint injury or overuse or altered mechanics may result in the production of certain degradative products, perhaps, that are targeted by IgE, or perhaps it's some allergen-associated reaction, but that the IgE basically traffics to the joint. We show, obviously, that depletion of peripheral serum IgE protects mice, but the IgE enters the joint, is coupled with the mast cells, and results in their activation, results in the production of tryptase, which degrades cartilage, as well as multiple other inflammatory and degradative mediators. And this kind of results in the progression of joint destruction and the breakdown of joint. So I just want to end with a couple kind of summary slides framing kind of how my lab views OA, and taking and adapting the models of others, thinking about how OA might develop. Perhaps there's certain predisposing factors, propagating factors that ultimately result in OA as joint failure. And, it's altered biomechanics and trauma, different types of injury, obesity, malalignment, repetitive use. And that these result in activation of the innate immune system, perhaps the complement system from fibromodulin release, perhaps certain inflammatory macrophage subtypes, and mast cells, as I exhibit in my presentation here. And this results in the production of various cytokines, chemokines, inflammatory mediators, and this basically promotes the pathogenesis of OA by promoting tissue destruction and remodeling, ultimately resulting in joint failure. So in my kind of view of OA, and I think increasingly the field, these different cells that are resident in these tissues that are involved become activated in these pathogenic ways and make inflammatory and degradative mediators. These damage cartilage and bone and the synovium, and this results in this cyclical process. And we're very interested in taking anti-inflammatory drugs and using them to target these pathways. Obviously, there's a lot of failures in OA. If you give an OA patient anti-TNF, our drug for rheumatoid, it doesn't work. If you give them, anti-IL-1, I think it's controversial what role that's actually playing, because there's discordant results in different trials. But I think perhaps we're targeting the wrong molecules. I think of rheumatoid arthritis and psoriasis and Crohn's disease, classical autoimmunity is like the house being on fire, flames coming out of the roof. I think osteoarthritis is fundamentally a different type of inflammatory response. It's an innate immune fire. It's like an electrical fire in the basement. And you're going to need a different type of fire extinguisher. You're going to need a different set of inflammatory drugs to effectively treat this innate immune inflammation that's driving OA. And so with that, I'll just recognize the people in my lab, Chen Wang and Erica von Kappler, and Heidi Wong really drove all these experiments. I collaborate with multiple people here at HSS. Bella and Susan are orchestrating a synovial histology and transcriptional analysis study that I'm heavily involved with. You know, Ed DiCarlo is obviously a key collaborator on the synovial pathology. And I, have been interacting with the NIH AMP, with Vivian Bykirk, Laura Donlin. Miguel is also involved in this large study with Bella, Susan, and Ed. And we collaborate with Dana on rheumatoid arthritis studies.